and we've got meiosis. So can you have a little think, take a second, can you write down a couple of facts that you know about mitotic division? What's important about it, or special or different to meiosis, and what it's used for? And then similarly, let's take a minute to have a little think about meiosis. How does it differ to mitosis, and what is it for? So mitosis is a cell division which is going to produce cells that are genetically identical. So there's no variation, there's no change to the genome. And so that makes it useful for the following. It can be used for growth. So I'm just increasing cell number. I don't need different types of cell. In fact, I need the same type of cell. I just need more of them if I'm growing a tissue. It can be used for repair. So if I damage some cells, those damaged cells need to be removed and need to be replaced. So I can do that with mitosis. And there is a sort of special case of asexual reproduction. And that's the reproduction that we see in things like bacteria. Meiosis then, different to mitosis because it's going to produce genetic variation. And it is the cell division that is used to produce our gametes. Remember, gametes is just a fancy term for the sex cells. The cells are going to join together in order to make a new organism. So we then need to know a little bit about asexual reproduction and a bit about sexual reproduction and the process. So we'll do sexual reproduction first. So we're going to fuse in this process. We're going to fuse, join together the nuclei of two gametes, so two sex cells. And the fusing together of the nuclei of two gametes forms a zygote. And that is a fancy term for a fertilized egg. So we fuse together in humans, our sperm and our egg cell, and they produce a fertilized egg, which is called a zygote. And this is essentially the process that is producing offspring. And they're going to be genetically different from each other and from the parent. In humans, the sex cells, or in mammals, sorry, are sperm and egg. And in plants, we have pollen and egg. So sexual reproduction is the process of joining two nuclei together from our gametes, producing a zygote, and that zygote is going to be genetically different. This is going to grow into a baby, into the offspring of the parent, and it's going to be genetically different to the parent. And then if you have more than one offspring, all of those offspring are going to be genetically different. In humans, we're going to join our sperm and our egg. Remember, these are made by the process of meiosis in the ovaries and the testes. And similarly, we're going to join together pollen and an egg to produce produce a new plant. And again, that is going to have variation because the process of forming the pollen in the egg is done by meiosis. So then we've got asexual reproduction. Asexual reproduction. In this case, we don't have any sex cells or fertilization involved. So no sex cells or fertilization. In fact, what we're going to do only requires one parent. And that parent, because there is only one parent, there's no fusion, so no joining of any nuclei, and no mixing of genetic information. So one parent, no fusion, no mixing of genetic information. And the process produces genetically identical offspring from one parent. Sorry for the interruption, chaps. Give me 1 minute. Okay, so we've got asexual reproduction, no sex cells or fertilization. We've got one parent, no fusion, no mixing with genetic information. It's going to produce genetically identical offspring from one parent. And the process, the only division happening is mitosis. So we need to know a little bit of detail about meiosis. Obviously, an amazing process that produces genetically different sex cells. And in fact, you're producing about 300 million sperm a day, and every single one of those sperm has a different mix of genetic information. And in fact, every single sperm that you ever produce in your lifetime is likely to have a different mix of genetic information. So let's do a little bit of detail on meiosis. A little subtitle, meiosis. So the cells in your reproductive organs are going to divide by meiosis, to form our gametes. The cells in your reproductive organs so our ovaries or our testes. Remember, in ovaries, it happens when the baby is in utero. So when the baby is still developing, that is when this process is happening. Girls don't actually produce any more eggs by meiosis after they're born. They're born with all the eggs they'll ever have, and then they start releasing those eggs in the menstrual cycle, the process of menstruation and the menstrual cycle, and that is part of homeostasis. But we'll go back to your ovaries and your testes, your cells in your reproductive organs divide by meiosis. To form your gametes. Remember, we're just fancy term for the sex cells, for those sperm and those eggs. And somewhere along the line, you're going to have to halve the number of chromosomes. Number of chromosomes is halved because we then need that sperm to join with an egg, and so that sperm needs to have 50% of the DNA and the egg needs to have the other 50% of the DNA. And so remember we call a normal somatic cell, so like a normal body of the cell, a somatic cell. That's the majority of the cells in your human body are diploid, which means they have 2N or 100% of the genome. Your gametes have half, so we call them haploid. They have N and they have 50% of the genome. And it's a random 50%. So there's a couple of stages at which some mixing can happen. So the best way to represent this is probably a diagram, and we'll pick some colors for the different types of chromosomes. So let's do a little diagram of the process. Now, if you go on to study A level, you'll need to know a bit more about this process, and certainly a bit more about the mitotic division as well. You learn all the different stages, but for now, all we need to do is just be able to represent, I guess, the DNA or the chromosomes at each stage of our division. So we'll draw a cell and we'll draw some chromosomes inside it. So here is the cell at the beginning. And we're going to pop a little nucleus in there. And we'll put some chromosomes. So we've got some chromosomes from mum. That's maternal chromosomes. And we've got a couple of chromosomes from dad. And we'll call those paternal chromosomes. And then this is your cell membrane, and this is your nuclear membrane here. So first thing we've got to do is we've actually got to duplicate the DNA before we go anywhere else. So let's just redraw our cell. Our nuclear membrane. And so we're going to duplicate our maternal chromosomes. And we're going to duplicate our paternal chromosomes. We then need to break down the nuclear membrane. And we're actually going to pair up the chromosomes along the center of the cell. So remember, this is not the right number of chromosomes. We're just doing a little representative sample. Okay, so there they are, nicely lined up. And it's at this point here that we can begin to get our first mix of DNA. So what happens is the arms of the chromosomes can actually swap over, and it's that swapping over that is going to mix our DNA. So let's represent that here. There we go, we've swapped a maternal arm onto there. And we've swapped a maternal arm over to there and a paternal arm on the other side. Should have a bit picked by the colours, shouldn't I? And again, a bit of a mix with our other chromosome. And so now you can see we've already got quite a good mix. We're going to do our first division. So division 1. And we're going to produce a couple of cells like this. And they are going to have inside them a bit of a mix already. So let's put this one and this one. And on the side, we'll have this one and this one. So we've now got some variation already. Sorry about the terrible drawing there. And now we go for division number 2. There's our second division. And we've got a real mix now. The chromosomes have been separated, and we've now got quite a mix of maternal and paternal DNA in them. And these cells are haploid at this point. They've got half the DNA required to make the organism. They've got half the genome. A couple like this. Yeah. And these are haploid. So let's go through it again from the top. And I'll add some labels to my drawing this time. So we've got first part of the process, our chromosomes are duplicated. Chromosomes duplicated. We've then got our chromosomes lining up in pairs. Chromosomes line up in pairs. And remember, we just here we add the nuclear membrane breakdown. and then we get our first division. So in the first division, each new cell has one copy of each chromosome. And then in the second division, I'm going to line up again. And in this division, the arms of the chromosomes are going to be pulled apart. The arms of the chromosomes are pulled apart. And you're creating 4 haploid daughter cells. So the result is 4 haploid daughter cells. This is a really important process because it increases our genetic variation. And that's really important, particularly when we go on to study natural selection. We know that it is this variation, along with mutations, that mean that firstly, we can evolve, but secondly, we're also less susceptible to diseases. An example of a counter to that is the identical bacteria. We know that bacteria are identical, and we can treat them therefore with one particular medicine, one particular antibiotic, and that will kill off all of those bacteria. That then leads into the problem of antibiotic resistance. One of those bacteria gets a mutation that means that they now vary from their identical cells around them, the bacterial cells around them, and they're more likely to survive, and that causes us a problem as well. So increases genetic variation, and we get sort of a random combination of maternal and paternal chromosomes. So the next little bit of the topic is just around your genome. Of the page. So the human genome, or just let's talk about the genome in general. So the genome is your entire set of genetic material. And we know the entire human genome. So we have looked at all of the genes within the human genome, and we're now able to identify what a large number of the genes do. We don't know what all of them do. We don't know what the combinations of them do, but we are able to identify certain genes with certain outcomes or certain phenotypes. So remember, you have your entire genome in every single cell in your body, apart from your sperm cells. They've just got that 50% of the DNA needed to make a human. So we need to know a little bit about the structure of the DNA. That is the molecule on which your genetic information is stored. A little subtitle. Structure of DNA. And DNA is just a chemical that has your genetic code on it. Remember, it stands for deoxyribonucleic nucleic acid. It's a polymer. And it's made of 2 strands that are wound in a double helix shape. When it is wound tightly, it forms your chromosomes. So the chromosomes are the structures that form when your DNA is wound tightly. And all of this is located in the nucleus of the cell. Within your DNA, you've then got genes, and those genes are little short lengths of DNA, and they code for a particular sequence of amino acids. So your genes are a short length of DNA. That codes for a particular sequence of amino acids. And what that means is those amino acids then join up and form different types of proteins. So I may have a gene for the protein that makes the enzyme amylase. So the gene will be the short length of DNA, and it's coding for different amino acids. Those amino acids are then joined together in a particular sequence, so they're floating around randomly, and a little mechanism reads your DNA and joins these amino acids together like little building blocks. Those amino acids are joined in a particular pattern, and then they will fold up and form your proteins. And those proteins are going to do a number of things. They're either going to be structural proteins themselves, such as collagen that's found in your skin, or maybe they're going to be enzymes, going to fold up and make enzymes, or they're going to fold up and make hormones. So we've done a lot of work on the human genome, and it's important to us in terms of a number of different uses. It has the research of the human genome has a number of different uses, so we do just need to know what they are. It's worth noting that actually, although we know the entire code of the human genome, we definitely don't know what it all does. There's huge portions of your genome that are non-coding sequences. There's these large, gap fillers that just don't seem to do anything, and a lot of them seem to be sort of maybe some inserts of some old viral infections or something. So there is still a huge amount of research that you can go away and do, and particularly if you're going into any kind of medical research, the likelihood is one of the starting points will be the human genome and genes, and if you turn genes on and off, what impact does it have for certain diseases? So it's still although we know the human genome, we know the entire sequence from the beginning of the first chromosome all the way to the end, and in fact you can see them all published in a book. We don't 100% know what all of it does, and so there's still a lot of research to do. So if you're interested in genetics or interested in sort of kind of medical research fields, then definitely going to be some interest in the human genome. So we completed that project in 2003, a couple of years ago, and it was an international collaboration of lots of labs working together to kind of piece together every gene so that we can have them all laid out in order. So the importance of this, I guess, from a medical perspective, helps us understand how certain genes are linked to diseases and genetic disorders. So how certain genes are linked to diseases and genetic disorders. And in fact, we go on to study a couple of these, don't we? We go on to study what happens if we damage some of these genes, or we mutate some of these genes. They cause some pretty significant genetic disorders. It has also been useful for us to study human migration. And we've certainly found a lot of Neanderthal DNA mixed in, which has then helped us track where humans have historically migrated from. And so these two are still very topical and still quite important areas of study that, again, you might want to think of going into. Okay, so we need to now look at, now that we know we've got a bit of structure, we've got some DNA. That DNA is has codes in it for different genes. Those genes are coding for a little list of amino acids. Those amino acids are joining together to form proteins, for instance, structural enzyme and hormones. So what we now need to know a little bit about is inheritance. So how we have inherited different characteristics from our parents, and how those characteristics are passed on. So we've got a couple of key terms. So I'm just going to do a list of some of the more complex terms, and I'm going to give you a couple of minutes just to try and write a definition. What's your understanding of the following terms? So I'm just going to write them here. I'm going to start with this one. Allele. We've got dominant. We've got recessive. We've got homozygous. We've got heterozygous. We've got genotype. And we've got phenotype. So we're going to use those words non-stop now. They're all quite unusual words. You absolutely need to know what they mean in order to understand the next bit of monohybrid inheritance. So just take a minute. What do you know about each of those? Can you just write down a couple of words? Okay, an allele is this. Dominant when referring to genetics means this. Recessive when referring to genetics means this. Homozygous, heterozygous, genotype, and phenotype. I'll just give you a couple of minutes to see how much of that you can get down. Okay, so remember, alleles are just different versions of a particular gene. So, for instance, you might have a gene for hair color, and you might have a brown allele, that version of that gene, or you might have a blonde allele, that version of that particular gene. Dominant is referring to the alleles. The dominant allele is always expressed. That means we always see the phenotype for that genotype. And that's even if we've only got one copy present. Recessive, so a recessive allele is only expressed if two copies are present, i.e. recessive allele is only expressed in the absence of a dominant allele. So a recessive allele is only expressed if two recessive alleles are present. Homozygous. So this is referring to the two alleles. Remember, you've got one from mum, one from dad. You might have one dominant and one recessive, or you might have two dominant, or you might have two recessive. So if you have two alleles that are the same, they are homozygous. So two alleles the same. And that means you can be homozygous dominant, or you can be homozygous recessive. So remember, dominant is often characterized by a capital letter. So for instance, homozygous dominant for the gene A would be that capital A, capital A. So this person is homozygous because they're the same. And in fact, we can describe them as homozygous dominant because we've got two alleles that are dominant and the same. And so equally, we could be homozygous recessive. So recessive is characterized by a lowercase letter. Remember, it's best to pick a letter where it is easy to see the difference between capital and lowercase. And in fact, in all examples, pick A, unless they tell you another letter in the exam, but you can just pick A. Is really clear which is dominant, this one, and which is recessive, this one. And this is homozygous recessive. So then there must be a name for if I have one dominant and one recessive, and sure enough, heterozygous means that you have two alleles that are different. And that is denoted by the following: capital A, lowercase a. So I'm not heterozygous, dominant, or recessive. That's not how we say it. You're just heterozygous. Heterozygous is this one genotype, one dominant, one recessive. You are therefore heterozygous. Your genotype then, your genotype is your combination of alleles. Combination of alleles. So if they're asking you to write the genotype, they're asking you to write this for a heterozygous person, or they're asking you to write this for a homozygous dominant, or this for a homozygous recessive. That is the person's genotype. And their phenotype, therefore, is how they look. This is the observable characteristic. Observable characteristic that is controlled by their genotype. So this would be my hair color based on whether I have two dominant brown hairs or two recessive blonde hairs, or a brown hair and a blonde hair. And sure enough, because I've got the dominant one, remember we said dominant is always expressed, and so my genotype would be heterozygous, and my phenotype would be that I have brown hair. Okay, so now we need to have a little go at using those terms to then track something called monohybrid inheritance. So at GCSE we do something called monohybrid inheritance, which is just the inheritance of a single gene. But let's have a little go at that. So another little subtitle: monohybrid inheritance. So in order to track monohybrid inheritance, the inheritance of alleles or a single gene, we can use something called a Punnett square. And the Punnett square is a really neat way of showing how, based on mum's genotype and dad's genotype, what the probability of different combinations is. What are the possible combinations of alleles for the offspring that those parents could have? And it can be laid out nice and neatly in the table, and then we can describe we can highlight the different genotypes, we can describe the phenotypes, and in fact we can give a ratio of the different combinations. So we could do an example. Let's start with a nice example for a pea plant. So we're going to do a pea plant. And they'll tell us, they'll give us a little bit of information about the pea plant. They'll say that the plant can be tall or short, and the tall allele is dominant. And they'll say choose a letter. We're going to choose T for tall, and the small allele for a short plant, small allele is recessive, and we'll do a lowercase t. And so they'll say to us, you're going to breed together two plants, and we're going to breed together some pure breed plants. So pure breed is a way of just saying that they are homozygous. So pure breed is homozygous. So if I say a pure breed tall and a pure breed small, so let's have a little go at that. So we're going to do pick our letter. So they might have given it to us in the question. They've given us a T, and then we're going to work out what the genotype is so that we can cross it in a genetic diagram. So we've got a pure breed tall. This is the phenotype. The genotype for pure breed tall must be homozygous dominant. And we're going to combine that with a pure breed small. And because it's small, therefore there must be no dominant allele, so it is homozygous recessive. And we can now put those together in a nice little cross. So here we go. So if we put one parent on the side here, remember this is the parent's alleles. So a somatic human cell would be that. And remember, during the division, it would produce cells with half the amount of DNA, and so they only have one copy of each chromosome, and then one different one of each of the alleles. So we can sort of imagine when we put this table together, what we're saying is these are the different types of sperm that will be created, and they can join together with different types of eggs. And so the other parent is tall, so they are homozygous dominant for T. And now we can cross that together. So again, this is a bit like this would be their somatic cell, and then that would be divided into two eggs, and we could join those together. So it could be pollen, couldn't it? This is a plant, so it could be pollen joining with the eggs. And so we can now combine those two. So this egg joins with this pollen, or this egg joins with this sperm and produces that genotype. Same here, same here, same here. And so what we know is 100% of the offspring are going to be heterozygous. heterozygous CT. And we also know that's their genotype. Remember, we also know their phenotype. They're going to be 100% tall. All of the offspring are going to be tall. Okay, so now let's take the question a step further. I'll let you do this one on your own. Now, what I'm going to do is plants. So inbreeding is not a problem. In fact, we do lots of inbreeding in plants. When we find a characteristic we like, that we know will sell well, we want to breed plants together to maintain that characteristic. So we're going to have a little go at that now. We're going to take two of these plants. So we're now going to breed together two heterozygous parents for the allele T. So can you now do a little Punnett cross of two parents that are heterozygous, and then see if you can see what the outcome is of that? I'll give you a couple of minutes to do that yourself. Okay, so remember, both of these parents are tall because they've got the dominant allele and it's being expressed over the recessive allele. And they're going to make some gametes that have the dominant and some gametes that have the recessive. So we're going to have some gametes that make the dominant and some gametes that make the recessive, and then we cross them together. So if these two join, it's going to make that. These two join, it's going to make that. These two join, it's going to make that. And if these two join, it's going to make that. So now, if we define just by height, we could say we've got three tall and one short, or we could separate it out into the different genotypes. So we've got 25% homozygous dominant. We've got 50% heterozygous. And we've got 25% homozygous recessive. I'll just try and highlight that with some colors. So here we go. This is one. That's that one, isn't it? And then we've got these two, these heterozygous ones. That's their 50% chance. And then finally, we've got 25% chance of homozygous recessive. Okay, we've got time for one more. Let's do one more cross. Let's have one more go. So this time we've got two parents. We're going to have one parent that is heterozygous. And I want you to cross that with one parent that is homozygous recessive. So again, you have a little go at the cross yourself. Try and lay it out like this. Tell me what the different outcomes would be. You can we could improve this, couldn't we? Mr. Foss could actually remember to write the genotypes. So there's the genotype there. There's the genotype there. There's the genotype there. You've probably laid it out more neatly than that. So let's have another go. Let's do a cross. I'll give you a couple of minutes to do that yourself. And when you get to A level, you get to do dihybrid crosses, which is significantly bigger than this. it's a good idea when you're writing your genotypes, put the dominant letter first. There's no rule, but it just looks better. It's the standard way of writing it. So you look more like a scientist if you're writing it this way. So just a couple more minutes to try that. You're crossing, remember, a heterozygous with a homozygous recessive. So you're picking the right genotypes for the parents. You're crossing those genotypes together correctly, and then you are saying this genotype is this likely, and it will produce this. This genotype is this likely, and it will produce this. Quite a common 4 or 5 mark question. So let's put our parents in. So I've got one parent that is heterozygous. There they are. And I've got one parent that is homozygous recessive. There they are. Let's cross them together. So I've produced someone that is heterozygous there. I've produced homozygous recessive there. I've produced heterozygous there. And I've produced homozygous recessive there. So then we know that this one is 50%. They are heterozygous. And they are tall. And we know that this one also 50% homozygous recessive. And we know that they are short or small. So a nice way. It's good to practice these. If it comes up, it's going to be a 4 or 5 marker. You're going to want to lay it out like this. They might make it slightly trickier. They might try and catch you out by talking about an initial. They might talk about this initial cross. They might not give it to you. They might say, we've bred together 2 pure breed plants. One was homozygous dominant and one was homozygous recessive. They may just describe this to you in the exam. They say, okay, well, now we're going to take 2 of the offspring and we're going to make them together. And so you need to, in your mind, say, well, I know that if I'm ever mating together 2 homozygous dominant and 2 homozygous recessive, I know it produces 100% heterozygous. So I know that the parents that they're asking me to join together are going to be heterozygous. I'm going to take those heterozygotes and I'm going to join them together. And I remember Mr. Frost saying it's going to come out 25, 50, 25. It is worth, I have rushed this a bit, I apologize. It is worth putting the phenotype. So that this is tall, and this is short, because you know it. These letters are giving a huge amount of information. You know it, so write it down. And there we go. So they might have initially said to us, we've taken 2 pure breed plants, homozygous recessive, homozygous dominant, tall and short. We bred them together. We took 2 of those offspring and we bred them together. What was the outcome? And they're looking for you to go straight to this. Okay, fantastically well done for joining on a Friday afternoon. As I say, I'm going to carry on with B2. In fact, we'll carry on with this next week. I'll just go straight into talk a little bit about family trees, and then we'll go straight into inherited disorders, and we'll talk a little bit more about this and how we can then track some different types of disorders. Well done for joining. As I say, take some time to have a nice rest this weekend, but make sure you're doing revision. Try and break it up into little chunks of revision. You really want to be working every evening and definitely every day of the weekend. Make sure you give yourself a rest, reward yourself for revising. Good luck with it all, and I'll see you all next week. Thank you very much.